Our next session is a panel, and just to make it clear, there's a workshop with the same participants. So we have 4 speakers in 45 minutes, which means this session is very tight, but you will have the opportunity, if you wish, to come to the workshop and ask them questions. So our first speaker is Nancy Ostiguy, who is an associate professor of entomology at Penn State University, with an interest in sustainable and organic agriculture. She has quite a few publications relevant to her studies, and she's currently working on the relationship between native bees and native plant distribution. I'm not going to go any further because I'd rather hear her than talk about the wonderful work she's doing.
Okay. So, just ever so briefly, my background is a little bit broader than entomology. My Ph.D. is actually in environmental toxicology. I did my work using epidemiological principles. I started off working with people and ended up working with honeybees and other native pollinators, or other pollinators that are native, since honeybees are not. So I have a strange perspective in some ways, especially concerning what my colleagues think. So this is the group that we're interested in. And the group that we really pay the most attention to is this group here, with honeybees being the primary group, and as I said, non-natives. A couple of very cool bumblebees. I find them amazingly fascinating. And of course, physicists who really don't know much biology decided at one point that they can't fly. They were wrong, of course. They just had to figure out how they could fly. This center group here are a bunch of other bees, which we have, I'm not quite sure how many in the United States. In Pennsylvania, one of the counts was 350 different species. These are solitaries. And one of the things that's interesting and sort of apropos of some of our costumes is that these guys, the males, actually do participate in pollination. They don't have stingers. These guys here are all females. So the males, their role in the species life cycle is for reproduction only. Again, they don't have stingers. So the costumes should be modified. It's a what? No, it doesn't work that way. Sorry. The other group that I've been beginning to do some work on, just because I think they're cool, are all of the various lepidopterans. And one thing, if you're interested in pollinators in general, that you really have to pay attention to with both the lepidopterans and your solitary bees is you need to provide habitat for nests if you're looking at the bees, and you need to provide food for the lepidopterans if you want to see the adults. And if you don't provide the food for the larvae, you won't see the adults. And there are also some of these lepidopterans that are not pollinators at all. So one of the fun ones is the Luna moth. And that adult does not eat. So if you want to see the Luna moth, you need to plant the food for the larva. Then I had to put in a plug, these 3 in groups here. Beetles are important pollinators. Mosquitoes, there are over 350 different mosquito species, 400 of which may be problematic to people, and even the Anopheles mosquito, which is the transmitter of malaria. Not all Anopheles mosquitoes bite humans. Not all Anopheles mosquitoes can actually transmit malaria. So when people start talking about eradicating mosquitoes, we would be interfering with a lot of other species and not doing anything to protect humans. And then, of course, we have flies. So lots of pollinators that we tend to not think about. Well, I wanted to give you an idea of what we know versus what we don't know. And other speakers have done similar kinds of things. So bees, hymenopterans in general, are critical for food plants, whereas our lepidopterans typically are not, but they're critical then for ecosystems. So you're not going to find people that do agricultural research really that really function or focus on lepidopterans. They're beginning, though, to look at non-bee species and realize how important they are. When we look at pesticides, one of the things that has come from some of the data that we've been putting together recently is that not all pesticides are equal. And in order to sort of achieve our eventual goal of working with nature rather than against it, so same thing that Dr. Lundgren was talking about, I absolutely agree with, that the goal, yes, is to eliminate pesticides, but ultimately to actually have a system that works with nature rather than against. There are different modes of action, and not all modes of action are going to be problematic. Some of them are problematic for us, some of them are problematic for insects, some of them are problematic for plants, because that's the focus. One of the things that we really need to be able to do, whoops, I did that wrong. Oh, I pushed the wrong one. Let's see. No, that's... Okay. Yeah, it's a good thing. Thank you. I pushed the up button, not the down button, and oh well. As has been mentioned, not all methods of application are equal. We did try, EPA blows it on occasion, and more than often than not, unfortunately, but they did try to reduce risks to non-target organisms by going to seed treatment. They didn't succeed. One of the classes I teach, I talk to the students a lot about unintended consequences. And when we don't think about what's going to happen if we do X, we end up someplace we didn't want to go, and we are really bad about reconsidering what we're doing. So we need to think about our methods of application and what are the consequences to those, and make sure that of the pesticides that we're going to currently use, which ones are going to be least problematic. And one of the things that's incredibly important for pollinators is looking at exposure. Another thing that is important is that, and this is something that EPA is doing purely for practical reasons, they're using honeybees as a surrogate for all bees. And I'll show you some data that says that we really can't do that because there are some instances where honeybees are more sensitive to the pesticides that we're looking at and some instances where they're less sensitive. One of the things that we are constantly doing is categorizing. Humans are great at categorizing. And one of the most important things in my mind is that we know what our words mean. So the general word that everybody uses is pesticides, and then we can talk about specific things, nematicides, fungicides, herbicides, insecticides. And the thing to keep in mind with all of these is these are really marketing tools. So you go and buy an insecticide, and people think it only impacts insects. You go and buy a rodenticide, and oh, it's only going to kill those rodents that you don't want. Well, that's wrong. It kills anything that's a mammal. So our rodenticides that we have available, one of the favorite ones for a long time was warfarin. Some of you may know people that are on warfarin for heart disease. The dose makes the poison. So smaller people, children, are more susceptible, not just because of their size, but also physiologically. And one that we don't think about at all as a pesticide are antibiotics. People are beginning to think about that now because we're beginning to think about the microbiome in our guts. Women probably have encountered it more often than men, that when we get an antibiotic to deal with a bladder infection, a lot of times you end up then with a yeast infection because you've killed off your microbiome. Other ways that we categorize chemicals are how they work. And really the point about all of this messiness, and I'll talk more about it in the workshop, is that, of course, we look at fungicides, herbicides, and our insecticides and miticides, and all these data have actually been put together for one purpose, and that is to more effectively use pesticides. So we know modes of action, what the target site is for various chemicals that we're looking at, because we wanted to make sure that as we use those chemicals, we don't expose our pest organism to a single pesticide too often. You want to expose them to multiple modes of action so you don't select for resistance by that particular mode of action. So the people that are interested in insect management or herbicide management or fungal management have gotten together and they've come up with lists that denote how various compounds work within organisms. Now, one of the things that's important is that just because we know, for instance, that imidacloprid is a nerve toxin, works against, is a nicotinic, what's called an AChR agonist, just because imidacloprid does this particular activity within an organism doesn't mean that that's the only mode of action. We only have that information because that's what the people that work with insecticides are interested in. So one thing that's important, we've categorized each of our insecticides, herbicides, or fungicides into particular modes of action, but they can have multiple modes of action in reality. We may or may not know about them. So in fungicides, we actually do have a category called multiple target sites. Chloranthionyl, which some of you have heard about, is one of those that is problematic. We also have a whole lot of things that we don't know how they work, so unknowns. Another thing, and this is a variable or a result of how we categorize our pesticides as fungicides, herbicides, or insecticides, is that we have modes of action that show up in both categories, but we don't have the same chemicals in both categories. So we only will list the inhibition of acetyl-CoA carboxylase. We will only list in the herbicides those things that we use as herbicides. We don't include the materials that have the same mode of action that we use as an insecticide. So even with our data that we have today, lumping our insecticides by mode of action or herbicides by mode of action is still extremely limited. One of the other things that I wanted to mention, and so we can have the same target site or mode of action irrespective of the kind of pesticide. We can also have a pesticide show up in multiple categories. So most of us know Bt as an insecticide. Well, it's also listed as a fungicide. So our chemicals can come up in multiple categories too. If we're going to look at risk, which is what we're very interested in, at least in the work that I'm doing with bees, we're interested in all kinds of information about the actual toxicity of the chemical. So we just literally need to know how toxic it is. As I said, we don't necessarily know this information, multiple modes of action. Most of what we have is information about death. We have the acute exposure, the acute outcome, and we have LD50s. We don't have information typically about larvae and eggs. This is what EPA has recently decided to focus on. We have most of our data are adults. As I've already said, some bees are more susceptible than other bees. And we tend to know extremely small amounts about non-bee species. Just to give you a very quick example, imidacloprid in this particular instance is less toxic, so the smaller number indicates the species that the chemical is more toxic. So imidacloprid is more toxic to bumblebees than it is to honeybees. But it's not always the case. So sometimes honeybees are more susceptible, sometimes your other species are more susceptible. Just to very briefly, and I'll talk more about this in the pollinator workshop, exposure can be extremely complicated. Most of the data that we have to date are snapshots, sort of like a cross-sectional study. Well, we have data now that tell us that pesticides, some pesticides, but not all, can be observed all season long. So multiple times we have found them in the same location, in the same colonies. So some of the herbicides show up consistently throughout the year, some of the fungicides, some of the insecticides. And an important thing to notice about the insecticides is the only neonic is chlorthianidin. The rest of these are acetylcholinesterase inhibitors, are old pesticides that people think we're not using much anymore. The same pesticides are found in multiple locations. In this particular study, we had 7 locations, and they're observed in multiple years, and we observe these data over multiple years. So atrazine shows up a lot. Okay, so that actually was it. I need to show you my last picture. Honeybee, and that, whoops, and that's the, I did it again. That's the wax that comes out at the end when they're then building their comb.
Warren Porter is one of our board members, a very active board member, professor of zoology and environmental toxicology at the University of Wisconsin-Madison. He's done some very exciting research in a variety of systems that look at commonly used combinations of agricultural chemicals. So,
thank you very much, Routt. My purpose here today is to explain the 1-2 punch on the immune system of a particular subgroup of GMO products, namely those that produce Bt toxins and those that have Roundup or glyphosate embedded in them. Let's see, I've never run this particular, how does this work? Okay. Okay. How do you make it, the slides go forward? Forward, let's try this. There we go, okay. Figured it out. I want to point out also that the immune system is connected to the neurological and the endocrine system, and so an impact on the immune system means impacts on those other 2 systems too, and I want to show you why that's true. Everything is interconnected in the biological world, including our own bodies and those of the animals and plants that share this planet with us. On the left side here, we have a healthy, if you will, system, and I'm not sure where this laser, I guess this is the laser pointer there. Whoops, holy smoke. What happened there? There we go. Yep. That's what I pushed, but it screwed everything up. I'm not going to bother with that, but I'll just kind of point. Let's see, I want to advance to also. There we go. Your molecular and cellular systems that comprise your body.
So right in the very center is your laser pointer right there, and then forward and backward there.
Okay, thank you.
Yeah.
There we go. Okay, good. I think maybe I'm pushing it too hard, that's why. Okay, mass, energy, and nutrients, that's what you had for breakfast, that's what you're going to have for lunch. That's running your cellular molecular systems. Your organ systems, like your central nervous system, your endocrine, and your immune systems are all interconnected. There are about 60 different molecules that are involved in the communication between these 3 systems. They talk to each other all the time. One of the things that we've shown is that chemicals as you get lower and lower and lower concentrations, start getting into the realm of our own physiology, because we function in the parts per trillion range in terms of concentrations of things that change the way our bodies work. And when you get down into that same physiological range that humans, or that animals work in, you get effects that you don't see at higher concentrations. Anyway, let's see here. I want to focus on the gut, because this is where a key part of the immune system works. About roughly, some scientists have estimated up to 80% of our immune function is tied to our normal gut function, and that is really where the 1-2 punch comes from, these two kinds of classes of genetically engineered foodstuffs. You can see that the lower levels here, the molecular cellular systems and organ systems, support individual level functions of reproduction, growth, and behavior. At the population level, birth rates, death rates, and social structure. And at the community level, relative species abundance, immigration, emigration, and other variables. And when we want to consider that there's also very important genetic expression things going on here. The genes control the functions here, the molecular and cellular systems, but it turns out that environmental contaminants have learned how to hack the code by shutting off genes, and so we have a whole field of epigenetics that I don't have time to talk about, but what it means is that it's been shown now that several environmental contaminants, pesticides included, are capable of being transmitted at changing that genetic expression, and that change in genetic expression becomes heritable over up to 4 generations down the line. And so if we are having the same kinds of things that's been shown in rats and mice, especially by Michael Skinner at Washington State, we're looking at a 100-year hit on our genome. So temporary, short-term kinds of things are important, but also that we really have to consider long-term impacts, and that's part of the concern here too. So the two subcategories that I want to briefly talk about are Roundup or glyphosate, in terms of these resistant soybeans that allow you to spray very large quantities of the pesticide on them, and it doesn't affect their growth. And then also BT corn, which produces toxins called BT toxins, Bacillus thuringiensis. These are put in, the genes for these proteins are put into the corn, and not only does the tissue of the corn have it and the corn kernels, but it turns out that, and you also apply atrazine, so you've got atrazine added in here, and atrazine has neurological, endocrine, and immune effects. But this BT toxin, and you probably can't see this very well way up there, but I'm going to show you a much bigger diagram, but basically it's produced in the corn cells. When it is eaten and consumed, the digestive enzymes activate proteins that form pores in the cell walls of the gut, and that causes the cells to deteriorate and rupture, and it causes death in caterpillars. That's the basic design for BT toxins. I also want to point out that these same plants that produce the BT toxins in their tissues also exude these kinds of toxins right into the soil to impact the soil microorganisms that Kristen talked about last night. She pointed out how important they were in soil health. So here we have a functional schematic cartoon diagram of the gut cross-section. And so here's the wall of the gut. Here are the gut contents down here. Outside of this, we've got the body cavity, the blood circulation. And if we get foreign proteins or other molecules out here, this stimulates the immune system to go into a chronic inflammation because it looks like a foreign infection here. That's the idea. So the gut is, of course, full of microbes of all kinds, and these bacteria have a functional pathway in them that allows them to synthesize a whole class of amino acids called aromatic amino acids. It's called the shikimate pathway, and we'll see why that's important in just a minute. So if we have an organism that consumes BT corn in the diet, with those poor proteins, what happens is you punch holes in the wall of the gut. Now all this stuff is leaking out into the body cavity. The body says, uh-oh, we've got a major infection here, and it begins to mount a major immune response. If glyphosate or Roundup happens to be in the diet as well, this produces a substance, or it inhibits this whole shikimate pathway and shuts it off. Now what all of a sudden happens is you've shifted the whole microbiome community in the gut, and you've eliminated a lot of your beneficial bacteria that are so important in immune function, and you shift the whole microbiome into a whole suite of very toxic producing bacterial organisms that provide and generate all kinds of problems in the gut. Glyphosate, by the way, has also been shown to have endocrine disruptor effects and neurological effects. Again, verifying the interconnection between all three of those systems. Here is what Judy Carman and her colleagues showed in 2013 in a title in the Journal of Organic Systems titled A Long-Term Toxicological Study on Pigs Fed a Combined Genetically Modified GMO Soy and a GM Maize Diet. Here are the linings of the stomach of the GM-fed pig versus control pig. And what we see here is a great deal of inflammation. This is expansion of blood vessels, separation of the cells that constitute the capillary structure. And what that does is it allows immune molecules to move through the walls of the capillary into the region and begin to try to respond to what is perceived as an infection. Okay. We could argue, is that happening in humans? And there was a paper published in PLOS1 in 2013 by a whole suite of people, including people from Johns Hopkins University, and the abstract here tells the story. Our bloodstream is considered to be an environment well separated from the outside world in the digestive tract. According to standard paradigm, large macromolecules consumed with food cannot pass directly into the circulatory system. During digestion, proteins and DNA are thought to be degraded into small constituents, amino acids, nucleic acids, respectively, and then absorbed by a complex active process and distributed to various parts of the body through the circulation. Here, based on the analysis of over 1,000 human samples from 4 different independent studies, we report evidence that meal-derived DNA fragments, which are large enough to carry complete genes, can avoid degradation and through an unknown mechanism enter the human circulation system. In one of the blood samples, the relative concentration of plant DNA is higher than the human DNA. So what appears to be happening is that the gut is being laced with holes, and those substances are getting into the circulation, generating chronic inflammation. What does chronic inflammation mean? Well, according to Dietert and Dietert at Cornell, who are very eminent people in this area, chronic inflammation, is associated with asthma and allergic diseases, autoimmunity, infectious diseases, ineffective vaccine responses, cancer, neurodegenerative disease, neurocognitive loss, cerebral palsy, atherosclerosis, hypertension, and male sterility. I don't know if you've watched what's been happening with CDC, but there's a lot of things going on right now. Thank you very much.
I have to say, one of these years, I'm going to convince Jay and the other staff to not put so much into one short session. So our next speaker is Tao Orion, who is a permaculture designer, teacher, homesteader, and mother in the Willamette Valley of Oregon. She's known here as author of Beyond the War on Invasive Species, a permaculture approach to ecosystem restoration. She holds a degree in agroecology and sustainable agriculture from UC Santa Cruz, and so on and so forth. She has a keen interest in integrating the disciplines of organic agriculture. We're really going to try and stay on schedule because we've got some workshops coming right away. So And this is sort of a complicated session, but.
Hi, everybody. So we've been talking a lot about agricultural use of pesticides, herbicides, and other types of pesticides today. And one of my research interests has been in the field of restoration and conservation and the proliferation of invasive species in ecosystems and the use of herbicide to treat plant invasive species and the use of other pesticides, including rodenticides, and other insecticides to treat non-plant invasive species. And the use of these chemicals is really widespread throughout many different types of ecosystems that we may not expect to find them in. This includes rangelands, forest, wetlands, riparian areas, all throughout North America and beyond. In New Zealand, the federal government there has undertaken a widespread application of 1080 to kill the invasive opossum species that's proliferating there. And so this is aerial spraying of a rodenticide that is very nonspecific and kills many different types of mammals and birds they're finding. And there's not a lot of really good data about the amount of herbicide in the US that's being used to treat invasive species, but it's a fairly significant number. In 2007, which is the last year that data were reported to the EPA, 89 million pounds of herbicide active ingredients were used in non-farm settings, including schools, roadsides, and government-owned property. Here's a picture of rangelands. This is a BLM, or Bureau of Land Management, managed acreage in eastern Oregon. So this is to treat the invasive cheatgrass. So there's some kind of larger scale applications of herbicides in settings where you really wouldn't expect them to be. And it's a concern for many people who use these lands recreationally, as well as for exposures for all of the creatures that live in these ecosystems. Many of them are home to endangered species, threatened and endangered species. Here's a list of herbicides that are currently approved for use on federally owned lands. And there's a lot of land that's operated by or managed by agencies like the Nature Conservancy, where many of these herbicides are also used to treat invasive species. It was a big surprise to me as I moved into the field of restoration that herbicides were kind of considered a first line of defense against invasive species, and that rather than being something that's considered a last resort or not used at all, many people consider their use kind of the primary thing that you have to use to treat this problem. And there's a lot of issues associated with this. One of the main ones is that herbicide active ingredients are just one part of the story for any compound that's being applied on your land, whether it's on agriculture or beyond. 50 to 98% of any formulation that you buy in a bottle is a so-called inert ingredient, which is considered not active, not biologically active towards the target pest that you're trying to eradicate, whether that's a plant or an insect. But these substances are not biologically inert. Many of them are listed as active ingredients within the scope of the EPA's pesticide registration, but because they don't act on the particular plant in that the context of the bottle of Roundup that you're buying, they're not considered active ingredients. And the type, the exact nature of these inert ingredients and the amount that's in any particular formulation is unknown. They're considered trade secrets. So when you buy a bottle of Roundup, I'm assuming people here don't buy bottles of Roundup, but when a consumer or a farmer or a land manager goes to a store to buy a bottle of herbicide, we don't know what's in it, they don't know what exactly is in it beyond the amount of active ingredient, in that case glyphosate, that's contained in that bottle. And it may be 2% glyphosate and 98% a bunch of other things. Many of these substances, there's been a few that have actually been scrutinized for their toxicology. One of them that's often found in a bottle of Roundup is polyethoxylated taloamine. It's an adjuvant. It actually makes the glyphosate penetrate plant cells more effectively. And it's been shown that Roundup is more toxic or has more potential for toxic effects than glyphosate alone. So when these adjuvants are added and into an herbicide formulation, which they usually are, the toxicity is enhanced in unknown ways because many of these substances are completely unregulated from the EPA pesticide registration standpoint. And land managers regularly mix herbicide active ingredients and adjuvants in tank mixes that they feel are appropriate to their sites. And it's kind of anybody's guess to a large degree, at least in the context of non-farm management, what anybody's using at any particular time. These combinations of herbicides have never been tested, and so we don't really know what's going on. It's also a concern. I came across a study from the University of California that was looking at land managers who had tank mixes, backpack sprayers on their backs. They were often applying up to 25 times the recommended amount of herbicide when they were out spraying invasive species. There's no real way to know exactly how much is going on, whether you're using the right amount. Many people in these restoration contexts are not licensed pesticide applicators, so they don't really even know what it is they are doing. They never received training on these kinds of things. So for me, as a permaculture designer and an organic land manager, I'm really looking for and offering opportunities for engaging in land management from an organic perspective. So how can we start to think about managing invasive species without herbicide or other pesticides in the case of animals or insects? Mollusks, zebra mussels are a big invasive species in the Great Lakes region. People are using all kinds of different molluscicides to treat them. And these are in waterways that people draw their drinking water from. So, as many of the other speakers have mentioned, it's really important that we start to think about whole systems design approaches to addressing some of the concerns presented by invasive species. Many of them are filling niches in ecosystems that we've created by our agricultural design methods, nutrient and sediment runoff leading to eutrophication of waterways, which then causes the proliferation of aquatic invasive species like water hyacinth or hydrilla. If we start to see the kind of chain of events or web of interrelationships that lead to the proliferation of invasive species, we can start to manage land in different ways, manage their populations in different ways, and encourage biodiversity through conscientious organic landscape management. And so we've been talking a lot about some of the bad stuff, but there are some glimmers of hope within the context of these things we've been mentioning. The EPA has just started to test for synergistic effects of pesticide active ingredients. That's helpful. The process is really slow, and we're not there yet. And the regulation of inert ingredients is becoming more and more of a on the radar of that regulatory agency. The hope is, that we don't have to use these substances at all, but in our current regulatory framework, that's what's going on. And as more and more people become aware of the potential exposures that are going on in the context of parks, natural areas, trails, forests, waterways, there's a lot of places where herbicides are being used where most people aren't even notified. But more and more people are becoming aware that these are an issue and are starting to develop organic approaches to their management. So that's somewhat hopeful, and that's my presentation for now.
I certainly would welcome everybody to attend the workshop, and because these are 4 great speakers, and I know you have lots of questions, and they would have a lot more they'd like to talk to you about. Our last speaker for this session is Paige Tomaselli, who is a senior attorney at the Center for Food Safety, where she works on law and policy related to genetically engineered crops, organic standards, factory farming, and other food safety issues. She co-wrote a chapter in the recently released CAFO reader, The Tragedy of Industrial Animal Farms, entitled Changing the Law, The Road to Reform. She has a JD degree from the Vermont Law School, where she was a member of the Environmental and Natural Resources Litigation Clinic, where as a member, she published an international comparative animal welfare article through the Animal Legal and Historical Center. So welcome, Paige. We're looking forward to
Hi, good morning. Okay, I'll try not to mess this up. A little disclaimer, I had a baby 4.5 months ago, and so I have a very scientific condition called mom brain. And I'm going to be using my notes a little bit because my brain just isn't quite back where it should be. I work at the Center for Food Safety, and I wanted to talk today about how we use environmental laws to challenge pesticides and genetically engineered crops. I'm going to give a very brief overview of the laws that we use, what I can do in 10 minutes. And in the workshop, we can get into more depth, and I was going to give a case study. So if you're interested, come to that. So the areas that CFS litigates are, we have manyfold. So first, as you probably all know, we litigate in the area of genetically engineered crops, and we've used environmental law to delay the introduction of certain genetically engineered crops. There's both pesticide promoting and pesticide producing, as was described earlier by Warren. These are challenges against the United States Department of Agriculture and to a lesser extent, Fish and Wildlife Service. We also litigate in the area of systemic pesticides. So this is harm to pollinators and harm to protected species. Oh, great, thank you. There you go. And these are challenges against the Environmental Protection Agency and again, to a lesser extent, the Fish and Wildlife Service. We also litigate in the area of increased use of toxic and synthetic inputs, mostly at FDA, but also USDA. And then we litigate to maintain the integrity of the organic program. That's something I'm going to talk about in a workshop this afternoon. So if you want to come to that, we're talking about 2 cases we have against USDA. So the National Environmental Policy Act is probably the first offense in any environmental challenge. It's a statute that basically any federal agency that is going to engage in a major federal action, so approving a timber sale, for instance, they need to study the environmental impacts of that major federal action. And the reason is that these impacts have to be produced to the public so that the public can comment on them. So it's an element of transparency, and so that the agency knows what it's getting into before it approves a project. The downfall or the setback of the National Environmental Policy Act, of course, is that it's a procedural statute, which means that the agency can study the project, but the statute can't force the agency not to approve the project. Even if there's going to be detrimental environmental impacts, the agency can still approve the project. The statute has worked for delaying the introduction of genetically engineered crops. It's worked for actually getting some prohibitions on the use of genetically engineered crops and some pesticides on national wildlife refuges. But again, it's just, it only delays a project, it can't stop it. The second statute is the Federal Insecticide, Fungicide, and Rodenticide Act. This is EPA's pesticide statute, and it is basically it's a risk-benefit statute. So EPA has to determine whether or not there's going to be any unreasonable adverse effects on the environment, and this is defined as an unreasonable risk to man or the environment, taking into account the economic, social, and environmental costs and benefits of the use of any pesticide. Additionally, there's a public notice requirement for a new pesticide or a new use, and there's a registration review portion of the law. But again, like I said, FIFRA is a risk-benefit statute, so the agency, it's a balancing act, and you can look at the risk of a pesticide, but it's balanced against the economic benefit or the perceived benefit to agriculture, and the agency gets deference on that decision, meaning the court was generally going to side with the agency's scientific opinion on whether or not that risk-benefit was met. Additionally, there's significant challenges as far as the lack of transparency. The public comment process only happens after a pesticide is approved, and so you're basically commenting on something that's after the fact. And a lot of this has to do with confidential business information. And there's agency delay tactics as well. The agency has been delaying in its Endangered Species Act analysis, which has been really challenging. And there's a lag in public research as well. The agency will defer to the research that they're getting from the pesticide companies and not the research from the public realm. The Endangered Species Act is our substantive statute. It can definitely stop a project in its tracks. The agency engaging in an action has a duty to ensure that any action authorized by the agency is not likely to jeopardize the continued existence of any threatened or endangered species or result in the destruction or adverse modification of the critical habitat of such species. It's a significantly strong statute. The Congress has tried to gut it a number of times, but we are using this particular statute to challenge some pesticides. Center for Food Safety is using it to challenge two pesticides, two neonicotinoid pesticides, clothianidin and thiamethoxam. We're in the middle of briefing and litigating that case right now for their harm to endangered species, certain endangered pollinators and other endangered species. The Center for Biological Diversity has a massive case with hundreds of pesticides involved for the delays that the Environmental Protection Agency has had as far as consulting with Fish and Wildlife Service about the impacts of these pesticides on thousands of species. And another statute is the Administrative Procedures Act. This is kind of the catch-all statute. It requires certain procedures in agency rulemaking and adjudication. And it's used also when there's no right of review under another statute. So this statute requires a certain amount of public notice and comment, so public participation in these major agency decisions. And so you can use the statute, for instance, to ensure that that public notice and comment, that transparency is being met. And we are currently doing that in two cases that I'll talk about this afternoon, challenges to the National Organic Program, one about rules of synthetic chemicals in organic production, and another about organic compost production. And the agency went ahead and made unilateral decisions to create rules without engaging the public in the comment process, despite the fact that the organic program is founded on the public process. And so we challenged them in federal court using the Administrative Procedures Act. And so that was my 10 minutes. And I'd like to talk to you in more detail about this stuff in one of the panels this afternoon.
Let's go ahead.